Welcome to the deep dive. Today we're tackling a really significant area of health, issues related to the growth of new blood vessels, that process called angiogenesis and neovascularization.
Right. Think of it as the body's plumbing repair crew.
Exactly. And when that system isn't working right, like we see in radiation-induced enteropathy,
Oh yeah, a serious complication from radiation therapy.
it causes major problems. And a key part of that is damage to the tiny blood vessels in the gut.
That microvasculature, it's absolutely vital.
Yeah, if that gets compromised, it really messes up the whole healing process.
It does. That network delivers nutrients, oxygen, takes away waste. Radiation damage there just starts a cascade of bad effects, really hinders recovery.
Which brings us to something pretty fascinating, photobiomodulation, or PBM.
Using light for therapy.
Yeah, it's non-invasive, uses specific light wavelengths, and it's showing, well, remarkable promise in speeding up wound healing.
And a crucial part of that is stimulating angiogenesis, right? Encouraging that blood vessel repair and growth.
Exactly. And what's really interesting, stem cells seem particularly responsive to PBM.
They do. They really seem to listen to that light stimulus.
So our mission today is to really get into the compelling science behind PBM. Specifically, how does it enhance angiogenesis? And could be a real solution for conditions where, blood vessel formation is impaired.
Sounds like a plan.
We'll look at some key research highlighting these positive impacts and also touch on how MedWave Group is applying this science, using specific wavelengths that fit right into this picture.
And it's compelling because PBM seems to enhance what mesenchymal stem cells, MSCs, already do naturally.
Right, those versatile repair cells.
Yeah, PBM can kind of give them a boost, specifically in promoting new blood vessel growth. That combination looks really powerful for things like that radiation enteropathy we mentioned.
Okay, so let's start there. How exactly does PBM interact with these MSCs? There are some really encouraging lab studies, the in vitro stuff. They found that treating MSCs with PBM, specifically at 1 joule per square centimeter, high intensity, it significantly increased their proliferation.
How quickly they multiply.
Yeah, at 24, 48, even 72 hours after the treatment. It was like giving them a go signal.
But the intensity seems key, doesn't it?
It does. The research showed that lower intensity PBM, much less effect on proliferation.
So getting the dose right, the parameters, is crucial for that biological response.
Definitely. And it wasn't just, more cells. They also saw increased mitochondrial activity inside the MSCs after PBM.
Ah, the powerhouses of the cell.
Exactly. You remember those from biology, right?
Yeah.
They handle energy production.
So PBM is basically giving the stem cells an energy kickstart.
Seems like it, at a really fundamental level.
And that enhanced mitochondrial activity is so important. More energy means the cell can fuel all sorts of processes division, making signaling molecules for healing.
And this boost seems to have, staying power. It wasn't just a quick burst. Longer-term studies, like 10 days out, showed that the PBM-treated MSCs formed more colonies.
The CFUF assay, right. That measures their long-term regenerative potential.
That's the one. So it suggests PBM has a lasting positive influence on their ability to self-renew.
That's a big deal. It indicates a real enhancement of that core stem cell property.
And what about the genes? What's happening there?
Good question. The research looked into that too.
And they found PBM treatment led to an increase in the activity of key stemness genes.
Like SOX2, NANOG, OCT4.
Yep, those are the ones. Think of them as master controllers, keeping the cells in that stem cell state, ready to develop.
So PBM doesn't just make them multiply, it helps them stay stem cells while doing it, which is critical for their therapeutic use.
Absolutely. Now, you always worry with stem cells, right?
Yeah.
Could the treatment accidentally change them in a bad way?
A very valid concern. You need them to stay stable.
Well, the research here suggests no. PBM treatment didn't seem to alter their basic identity.
How did they check that?
They looked at the surface markers, the proteins on the cell surface that define them as MSCs.
The immunophenotype.
Right. They checked the positive markers like CD44, CD90, CD105, and the negative ones they shouldn't have like CD34, CD45.
Oh.
And the PBM-treated cells looked highly similar to the controls. PBM enhanced them without messing with their fundamental profile.
That's crucial. Maintaining those markers means they keep their identity and are less likely to cause, say, an immune reaction.
And there's more good news on that front. Their ability to differentiate to turn into other cell types like fat or bone cells.
Did PBM affect that?
Didn't seem to.
Yeah.
Even with daily PBM over 14 days, the MSCs kept their potential to differentiate normally.
Okay.
They even check the gene activity for fat and bone development markers, PPRO, LPL, ALP, BGLAP, similar levels between the treated and untreated groups.
So PDM seems to boost the good stuff proliferation, stemness, without limiting their flexibility to contribute to repair later on.
Exactly. It's enhancing, not compromising.
That's a very important balance for therapeutic applications.
Okay, so PBM gives MSCs a significant leg up. But how does that actually help with angiogenesis, with protecting those endothelial cells lining the blood vessels?
Right, making that connection.
Well, the research looked at that too.
Yeah.
First, they confirmed that radiation does impair angiogenesis in human endothelial cells, HUVECs in this case.
Those umbilical vein cells. Yeah, in lab assays, radiation stops them forming those tube-like structures well.
Right, they just don't grow properly after being irradiated.
And that's a key part of the damage in radiation enteropathy, that impaired repair capacity.
But here's where the PBM-boosted MSCs come back in. Researchers collected the signaling molecules released by these PBM-treated MSCs.
The conditioned medium.
Exactly. And they put that onto the irradiated endothelial cells.
And what happened?
A significant increase in the tube formation, more total length, more branching points in those damaged HUVECs.
Wow. So the signals from the PBM MSCs were telling the damaged vessel cells to get growing again.
It looks like it, like they were sending out potent regenerative signals.
That's the paracrine effect we talk about with MSCs releasing factors that influence neighbors. PBM seems to amplify that.
Totally. It's like turning up the volume on those healing signals.
And did it just help them grow, or did it protect them too?
Both. The conditioned medium from the PBM preconditioned MSCs also protected the HUVECs from radiation-induced apoptosis.
Programmed cell death.
Right. They saw fewer dying endothelial cells compared to using medium from regular MSCs.
So it's a double whammy, promoting new growth and protecting what's already there from dying off.
Exactly. Which is huge when you're dealing with radiation damage that threatens both.
Makes perfect sense.
And digging into the why, they looked at the genes again in the MSCs after PBM.
And found?
Upregulation of a whole panel of pro-angiogenic genes. Things like VGF, HGF, BFGF, PDGF, ANGPT1, ANGPT2, SDF1.
The usual suspects for blood vessel growth. Key players.
For sure. And they didn't just see the gene activity increase, they measured the actual proteins. BEGF and BFGF levels were significantly higher in the conditioned medium from the PBM-treated MSCs.
So the proof is in the pudding, or the medium in this case. PBM really does boost the production and release of these crucial growth factors.
It certainly seems that way.
And those factors do everything from stimulating endothelial cells to multiply and move to stabilizing the new vessels. Very supportive evidence.
Okay, so the lab stuff looks really strong. But does it work in, a living system?
The crucial question, in vivo evidence.
Right. So they used a mouse model for radiation-induced enteropathy, irradiated the mice, then treated them.
With what groups?
They had a control group, a group getting regular MSCs, and a group getting the MSCs pre-treated with PBM.
Okay, the real tests.
And the results, really encouraging. The mice getting the PBM preconditioned MSCs did significantly better.
How so?
Less mucosal destruction in the intestine, better proliferation of the crypt cells needed for regeneration.
Measured by Chi-67 staining, probably?
Yep. And improved epithelial barrier function, too.
That's huge. It shows the lab benefits can actually translate to improving the overall condition of the irradiated gut in a living animal.
And maybe most importantly for our discussion today.
The blood vessels.
Exactly. The PBM preconditioned MSC treatment significantly reduced the loss of those microvascular endothelial cells in the irradiated gut lining. It helped preserve that delicate network.
That is critical. Protecting the microvasculature is key to recovery.
And they didn't just see less loss, they saw more growth. A significant increase in actual angiogenesis, new blood vessel formation in the tissue layer called the lamina propria.
They likely use markers like CD31 and CD105 to confirm that?
That's right, double positive cells, more new vessels forming in the PBM MSC group.
So protecting the existing vessels and promoting new ones, that really explains how they can alleviate the enteropathy, supports the whole healing process.
It really ties it together. The paracrine effect of MSCs, turbocharged by PBM preconditioning, makes a real difference in reducing that intestinal damage.
Yeah, PBM seems like a powerful way to modulate MSC function, boosting their natural repair capabilities by ramping up those crucial angiogenic signals.
Now, I want to quickly touch on another angle related to angiogenesis in PBM involving parasites.
Ah, yes, the support cells wrapped around capillaries. Very important for vessel stability and function.
Exactly. There was another study, this one looking at skin wound healing in mice. They used special mice where parasites were labeled.
Transgenic mice. Okay, so they could track them.
Right. They created 2 wounds on the back. One got PBM treatment, 660 nanometer light this time, 5 joules per square centimeter. The other was a control.
And what did they track?
They looked early on 12, 36, 72 hours at cell mobilization. They saw more undifferentiated cells, marked by nestin, moving into the PBM-treated wound.
Nestin-positive cells, often associated with stem or progenitor cells.
And interestingly, a specific potent subpopulation of parasites called type 2 parasites, which were nestin and NG2 positive, were also significantly increased in the PBM wounds early on.
So PBM seems to be calling in not just general repair cells, but specific parasite populations known to be involved in vessel formation and stabilization.
Looks like it. They also saw significantly increased blood flow in the PBM-treated wounds using Doppler analysis.
More flow makes sense if you're building and supporting new vessels.
And the histology backed it up. The tissue sections showed markedly better vascular supply and just more advanced tissue repair stages in the PBM group.
So PPM isn't just working via MSCs releasing factors. It might also be directly mobilizing crucial vascular support cells like parasites, another mechanism contributing to better healing and angiogenesis.
It seems plausible. Multiple pathways converging.
Which brings us back to MedWave Group's approach.
Right, this compelling research we've been discussing, showing PBM stimulating angiogenesis through MSCs, maybe parasites releasing growth factors. It directly supports what MedWave Group is doing.
Using those specific wavelengths, 635 nanometers and 810 nanometers.
Exactly. Those fall squarely within that red to near infrared spectrum that's been so well studied for PBM's beneficial effects.
Absolutely. That 600 to 1,000 nanometer range is known to penetrate tissue well and interact with cellular components like mitochondria.
Those powerhouses again.
Yep, stimulating energy production, reducing oxidative stress, and triggering the release of those growth factors we talked about, VEGF, BFGF, and others crucial for healing and angiogenesis.
So the science is really consistent here. The positive research clearly shows PBM in this red near infrared range is a valuable tool.
No doubt.
And MedWave Group is essentially leveraging that strong scientific foundation, applying these specific effective wavelengths to tackle health challenges where boosting blood vessel formation is needed.
It's about translating that fundamental science into practical applications that could potentially help people.
Makes total sense. The evidence we've walked through today really does strongly support the potential of PBM, especially with these types of wavelengths, to positively influence angiogenesis and neovascularization.
It offers a really promising, non-invasive avenue for a range of medical needs.
Okay, so let's try and sum this up. The key takeaway seems to be that photobiomodulation shows, well, significant promise for enhancing angiogenesis.
Definitely, through various mechanisms.
Yeah, boosting MSC function, triggering growth factor release, protecting endothelial cells, maybe even mobilizing parasites.
It's quite multifaceted.
And it all points to a very compelling approach for health issues marked by impaired blood vessel formation.
The research is positive, it's compelling, and it really highlights the potential role of PBM in regenerative medicine. Plus, it's non-invasive.
That's a huge thanks for taking this deep dive with us today.